So in this video, I'm going to go through how you would design cloning primers if you're doing classical restriction cloning. So I'm not going to talk about primer design in general. What I'm going to specifically talk about is the sorts of primers that you need if you're going to do classical cloning via restriction enzymes. So just to remind you of what that process is, there are other videos that explain this fully, but we've got our plasmid vector with an origin of replication, and we've got a marker gene in there. And then at some point in the vector, we've got a multiple cloning site, which is a whole bunch of restriction sites next to each other in the vector, so you can choose which one you want to use for your gene of interest. As I say, there's a whole other video there. So what we do is when we're trying to clone our gene of interest, so we need a copy of the gene of interest, and at the end of it, we add restriction sites which we do during the PCR process via the primer sequence. Okay, so we might add EcoR1 at the end and BAM-H1 at the end. It depends which enzymes you need to use, and there's a whole other video about how you choose which enzymes to use in your process. So let's just have a little bit of a reminder of how PCR works and what the contribution of the primers is to this process. So I'm just going to draw out a cartoon of the gene. So here's a gene I'm wanting to amplify. So the gene sequence that I want to amplify is in black, and then obviously that's connected to the rest of the genome, which is in grey. Okay, so the gene of interest is this region here. Okay, so if I was doing regular PCR, then I would design my primers like this. So I would have a forward primer at the start of the gene, which binds to the sense strand, so the top strand, and I'd have a reverse primer that binds on the opposite strand. And PCR primers, we always write them as thinking about going from 5' to 3'. So you may know that DNA polymerases are directional. Okay, so they can only work in one direction, and they have to work 5' to 3'. So we write our primers. 5' is where the polymerase will start, and it moves in the 3' direction. And then when that amplifies, what you end up with is, so you start with your primer sequence, and then the polymerase reads through, and then you get the rest of the DNA amplified, and in the same direction you have your DNA like this. So then obviously they'd be base paired together, so that gives you your PCR product. So the sequence of these primers ends up defining the beginnings and ends of your sequence. Okay, so regular PCR, you just have your primers, and each primer is 15 to 25 base pairs long, and that will be sequence that's unique to that particular gene. Okay, so you got 15 to 25 base pairs for the forward, 15 to 25 base pairs for the reverse. That means the chances of your PCR primers binding to a non-target gene is relatively low, although you do have to check it. And your PCR product is defined by the beginnings and ends of those primers. If we're doing cloning, then we need to adapt this situation a little bit in order to add those restriction sites. So I'll draw it again. There's my gene of interest in black. Okay, and then I'll just put it in dotted, just so it's a little bit clearer to see what's going on. Okay, so this is the DNA in the genome. Okay, and the black is the gene of interest that I'm trying to clone. Okay, so the cloning primer, it's similar, but then we've got a little bit of a modification. So we definitely do still need bits of the primer that are complementary to the gene of interest. So again, those bits will be 15 to 20 base pairs long. So that's got that matching sequence. So we definitely need ones that are complementary to the gene of interest. But then what we do is we add some extra stuff to the primer. Okay, so the purple equals the sequence of the GOI, and the blue bit equals things that we've added artificially. So these aren't in the natural sequence of the gene. You'll see that these primers aren't annealing to the gene. They're kind of hanging off the ends. Okay. Again, these are 5' to 3'. So the bits that we add artificially end up being added to the 5' end of the primer. The 3' end is the same. Okay. So when we go through the PCR process, what we'll get is we'll end up with sequence that we've got the primers and we've got the add-ons, and then we've got the rest of the DNA that's been made. Again, obviously that's all base paired together. So that's my PCR product. Okay, so if we have added a restriction site via these blue bits, these artificial bits of sequence, then our enzymes will be able to cut and we'll be able to put it into the multiple cloning site. Okay, so with our cloning primers, they're longer primers, so we still need the 15 to 25 base pairs bit of the gene, but then the artificial bit can be another 15 to 20 base pairs long, depending on what you're adding. You kind of want primer maximum is sort of 30 to 35 base pairs. So it slightly depends what you're doing as to whether you can fit everything out in that you need. Because you need 15 to 25 base pairs as your sequence, and you need some added stuff at the 5' end. Okay. So let's have a think about what we need to do. So I've got the sequence of a gene here. Okay, so again, the black is the gene I'm trying to amplify. I've not shown the bit in the middle, and the grey is just the rest of the genome. Okay, just going to highlight a couple of features in here. Okay, so let's do these in green. So the green is the start codon for translation. Okay, so we need to start with an ATG at the beginning. So this is defining the beginning of the protein coding bit of the gene. Okay, so that's the start codon there. And then here, that is a stop codon. So that's going to define the end of the protein. Okay, so when we make a molecule of mRNA, the ribosome will go along, it will find the first ATG, it will start translating, it will keep going until it finds a stop codon. So in this case, it's a TAG, is one of the stop codons. And at that point, it will stop translating, and that defines the end of your protein. Okay, so those are two quite important features to be aware of. So let's start with the forwards primer. Forwards is a little bit easier than reverse. Okay, so what do I need in my primer? Well, my primers need to have several different elements. Okay, so they do need the sequence of the G of interest. So we definitely need to include these bits in the primer. So the primer sequence will just be the same as the sequence along the top to make a new copy of it. So we definitely need the CAA, CGG, TA, CCG. It might need to be a little bit longer than that in order to get enough of the gene of interest. Okay, so we definitely need the GOI sequence. We definitely need, in this case, the ATG start codon if we're in the forwards, okay, because we need to have that so we know where the ribosome is going to start. So that is the start codon. That is definitely in my primer. Okay, I then need to have the sequence of the restriction site that I want to add. Okay, so different restriction enzymes have different sequences. So let's imagine that we want to add an Echo R1 site. Okay, so the sequence for Echo R1 is GAATTC. So now you can see this primer. This is no longer complementary here. It's just hanging off the end, but that's absolutely fine because we've got the 3' end to anchor it onto the gene. So that is the restriction site. And in this case is Echo R1. Okay. Then the other thing that we need is a leader sequence, which is 6 to 10 base pairs. And that can be any sequence you like. Okay, so I'm just going to put it as X's. Okay, so it could just be A's, it could just be 80, 80, 80, 80. Doesn't really matter. What that leader is there for, and that becomes the 5' end of the primer, okay? What that leader is there for is to trick the restriction enzyme into cutting your DNA. So restriction enzymes are what we call endonucleases. Which means that they cut within a DNA molecule. They can't cut at the ends. Okay, so they think they need to cut in the middle of a sequence. So what this leader is here for is when that's been amplified, we've got a little bit of sequence at the end. That's just enough to trick the restriction enzyme into thinking this sequence is in the middle of the piece of DNA, so it will cut it. So you have to have that leader sequence, otherwise your restriction enzymes won't work. Okay, so my forwards primer, if I start with a 5' end, so I've got my leader sequence, that might just be 80, 80, 80, 80. I've got my restriction site, so in this case it's 6 base pairs, GAATTC. I've got my start codon, and then I've got my sequence of my gene of interest. So that's my forwards cloning primer. Okay. So there's the forwards primer. Now let's think about the reverse. Now the reverse is a little bit more brain bending, okay, but let's go through it. The principle is exactly the same, okay. So let's work back through it. So we need the GOI sequence, okay, so in the reverse primer, so the forwards primer, we need to use the top strand, the sense strand. For the reverse primer, we need to use the opposite strand. We have to use the bottom or the complementary or the anti-sense strand in order to make our reverse primer. So we definitely need the sequence of the gene of interest. So we've got C, G, G, C, G, G, T, T, T in this case. And this will again be the 3' end of the primer because it's going in the other direction. Okay, so the GOI sequence is always at the 3' end. Okay. Then, if in a forwards primer we need the ATG, we might need the stop in the reverse. Okay, but we might not do. I'll show you why we might not need it in a minute. Okay. So let's assume we do need the stop codon, okay? So we do want the primer to end there. So we will need to include this ATC. That's the stop codon. Okay, so that will mean that when we express it, the protein will end there. Okay. We've then got our restriction site. So let's put in a different restriction site. Let's put in, let's go for CTC GAG, which is the restriction site for the ZO1 enzyme. Okay, and then we need a leader at the end of it. So again, 6 to 10 base pairs, really doesn't matter what they are, as long as they're there to extend it so that ZO1 thinks it's cutting in the middle. And there's our 5' end. Okay, so that's our reverse primer. Drawing it out against the DNA sequence. But when you're ordering your primers, you always order them writing it as 5' to 3'. Okay, so if I reverse that, I'll just do it at the bottom here. To order the primer, you go 5' to start with. Okay, so it would be X, 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 X. Okay, then it would be G, A, G, C, T, C. Then it would be, so I'm working backwards. Then it would be C, T, A, T, 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 G, G, C, G, G, C at the 3' end. So to order it, we have to reverse it, but it is the same thing as before. So that is your ZO1 site. Then the CTA is your stop, and then this bit is your gene of interest. Okay, that was the leader. So when you're writing out your primer sequences to actually order them, you have to flip them around. So you start with a 5', so you need to kind of read backwards along that way. Okay. Now, there is another version of making your reverse primer, which is if you need to remove the stop codon. So if you look at my protein tagging video, you'll find that quite often what we need to do is to actually remove the stop codon at the end of a protein, so that it reads into it. The ribosome keeps going and it reads into a second protein. Okay, which means that we have to change the sequences a little bit. So I'm just going to draw this out again. I'm just going to move it across. So T-A-G-A-A. I won't bother with the rest of it. So that's T-T-T-A-T-C. Okay, so the TAG, so this is in the genome, that is the stop codon. Okay, so if I want to remove that stop codon because I want to add green fluorescent protein or a protein tag on it, then I need to be a little bit different with my primer. So I'll still have this 3' bit, the sequence of the gene of interest, so that will end, it'll be exactly as it was there, just with the T, T, T. Okay, I'm still going to need my ZO1 site, CTCGAG, and I'm still going to need that leader. Okay, but I'm going to mutate this stop codon. Okay, so rather than having the sequence of the stop codon, I'm going to replace it with something else. So rather than having ATC, which will be the complement, I'm going to replace that with A. Okay, and that will removes the stop codon. Okay, so AAA, when that's in the protein, because obviously it's the complement, so AAA would become TTT in the protein, and that's a phenylalanine residue, so that's a regular amino acid. So we wouldn't have that stop, so the protein would read, instead of being a stop codon there, it would mutate, so it reads TTT, and therefore we get read through of our protein. Okay, so because we're putting in the mutation via the primer, all the copies that will have it will not have the original sequence, will have the change sequence. So although there is a stop codon naturally in the gene, because we've mixed about with it with our primers, so we have eliminated, in this case, we've eliminated the stop codon via the primers, so that means that there won't be a stop codon there either. So the protein will be able to read through. Okay, so you'll probably want to have a look at the protein tagging video to make full sense of that. Okay, so this is the reverse primer. Removing the stop codon. Okay, so that will be our reverse primer in that case to remove the stop codon. So in that case, just to order it, it will be 5' X X X X X X X X X as before, GAGCTC as before, but instead I'm going to put AAA in there. Then I'm going to go TTT, GGC, GGC, Okay, so that one is with the stop, and this one is no stop codon, because I've mutated that stop codon bit there. Okay, so there's no longer a stop codon, there's just a regular amino acid at that AAA. So that means that my protein, when I make it, it won't stop there, it will keep going on and then might go and express a tag, for example. Okay, so what do your primers need? This is the important bit down here. Okay, they need leader sequence at the 5' end. They then need your restriction site. Okay, they then need either your ATG if you're going forwards, or your stop reverse. And I'll just put a question mark there. Do you want a stop codon or not? Depends what you're doing as to whether you want to include the stop codon or not. Then you need your GNOI sequence at the 3' end. And if you do that, when you amplify, all of your PCR products will have the sequence that was in your primers, not in the original gene. So that means that you can add your restriction sites, you can add any tags or anything that you want via your PCR primers, so that can then go into your vector by the multiple cloning site.